Hey, I think I got into it. So you just mute the volume on YouTube Live.
Who, me?
All I see is a green screen on my YouTube Live. You see? Okay. Yeah. All right. I think we're live on YouTube right now, Rob, but I need to get my video fixed here. You still on Zoom?
Yeah, I'm still on the Zoom. Is this where you want me?
Okay. We're ready to go, Rob.
Nice.
Let me get resituated here. All right, Rob. Well, we are on here. Hopefully, guys will get switched over. A little technical difficulty there. It's usually why I stay out of technology. Um, Anyways, yes, Blake Merritt here, Core Bioag. We have Rob Shortell on the line and talking about amino acids, kelps, different things like that. And last night I talked to a guy and he called up and he knows about this video and we were talking and I was trying to explain to him what direction I wanted the video to go. And he looks at her, he says to me, he's like, "Well, you guys are starting in level 3." And he's like, "You're gonna just talk over everybody's head." And I was like, "Okay, that's a good point." So I started thinking about that last night, and I was like, I think we need to get back to the basics of what we're doing and what we're trying to achieve. And that goes back to talking about amino acids and what they are and what we can do with them and how to use them. And that's what I appreciate about you, Rob. And hopefully you can do some explaining on that. I know you've helped me quite a bit. And I think that's where we want to kind of start with this kind of introductory video is talking about what they are and why we're using them. And then we can get into a little bit about when to use them and then all of that stuff. But yeah, let's get a little introductory about you, Rob, and kind of what your background is, how you got into some of this, and kind of kick it off from there.
Nice. Well, you hear me okay over there?
Yep.
Great. Well, thanks for having me, Blake. It's good working with you. I'm glad we've been able to come up with such synergies between the two of us. And I think today I'm a plant physiologist by training. And today I want to talk to you about crop production and just going from the seed to the actual plant cell to the yield that we harvest and we take to market. So I'm here. I'm with Ag America. My name's Robert Shortell, I'm currently the president and CEO of Ag America. I'm all things plants. I've been a lifelong naturalist and a plant coach for many years. I have a BS in plant biology, a MS in weed science, and I got my PhD in plant breeding and genetics for environmental stress tolerance in grasses. My specialty is really the plant hormone environmental interactions, but more importantly, on managing those interactions in the real world and turn them into profitable acres. I've spent a lot of time in between academia and now the corporate world. We have multiple patents and lots of publications and things like that. And currently, Agmerica is serving farmers in 72 different countries and 49 states. So I'm looking to get up to 75 countries this year and make a trip to Alaska. So if you know any farmers up that way, that'll finish it off. But yeah, lately, at least the past 5 years or so, we've found a real place in some of the new practices with regenerative agriculture and looking at ways to grow crops more efficiently and maintain our yields with less inputs. So it comes right into my wheelhouse, and we've been having a lot of fun in the field.
Yeah, no, that's great. Um, yeah, I've known you here for for at least the last year now, and yeah, you've really taught me a lot on amino acids. Yeah, I've been familiar with amino acids. I think a lot of farmers out there maybe have an understanding of amino acids and what they can do from a biological sense, feeding biology, and how we can kind of grow that side of things. Um, but what I didn't know was the interaction that they had with hormones and how we could really target the plant growth stage and really understand how to use that amino acid properly and the implications that could bring to crop growth and all of that. And I think that's definitely what you've taught me. And every time we talk, I learned something more, and they seem to be really long conversations. And that's kind of what's brought us here to doing this video. What we did a couple of weeks ago and had a great turnout, and guys wanted more information. And so that's what we can hopefully bring to them here. But yeah, let's get started with some of the information you have. I mean, what are amino acids? Where do we derive these materials from? Basic understanding would be fish materials, whether it's liquid fish. I think you can get into some plant protein type amino acids. But kind of give us your take on amino acids, what they are, and why we're using them. What's the role of these amino acids, and what can they really do for us from a crop growth perspective?
Yeah, that's that's interesting because that's how we met. Was I guess that's where my world crossed into yours. We were just working with the different amino acids and hormone products, trying to increase yield and reduce environmental stress. And we happened to have a microscope with us, so we noticed that it was making real big differences to the soil. And that's when I linked up with the lab we work with, and then they introduced me to you. But amino acids are really cool. I mean, from the plant's perspective too, not just the soil. If you think about it, they're efficiency enhancers. They've already the sun hits the leaves, photosynthesis happens. We take CO2 from the air, and then the plant attaches that carbon to nitrogen to make the rest of the plant, grow and develop. So what it really is it's capturing that solar energy and it's converting it into usable forms of nitrogen. Excuse me. But the amino acids, they're really a lot of them are hormone precursors, and they're functioning in environmental stress response and just overall environmental or response to the environment in terms of growth. Some of the amino acids can work to increase stress tolerance in the plant, and they're also part of the regular nutrient metabolism cycle. So you get a lot of nutrient use efficiency effects coming directly from the amino acids. Some are proven to increase micronutrient uptake. Others can directly increase protein. And then even more, some of them are the direct precursors to different hormones, like tryptophan, for instance. That's the precursor to auxin directly. So in general, they're really important in all things plant growth. And in a crop where we're trying to fertilize the plants and then convert that nitrogen to yield, they are the direct intermediate between the nitrogen you apply and the yield you harvest.
Yeah, no, that's great. Um, Sorry, I was trying to get a little things figured out there on the the faulty Zoom meeting that we had.
Yeah,
Um,
yeah, no problem. I know this is their first in a series, so we'll have a little bit of technical difficulties. But my previous or my background is really in the study of hormones and how the hormones are the brains of the plant and how they lead from when you plant the seed to when you harvest the seed. And it turns out that the hormones are making the decisions as to what the plant should produce and whether or not it's going to produce amino acids that lead into protein and other stress responses and things like that, or whether or not it'll produce starch and cellulose or even lignin and things that lead to bigger plants that can harvest more carbon and capture more sun.
so going back to what are amino acids? A lot of times when you see amino acid material, they'll have an L in front of it. What does the L amino acid mean? Are there other amino acids out there, and what are the advantages of either one? Give us a little bit of background on that.
Yeah, there's lots of amino acids, particularly in the market, but in life, there'll be 20 different amino acids. So as I was kind of saying before, the CO2 comes into the leaf, mixes with the nitrogen. It's powered by the sun through a process called photosynthesis, and it's directed by the hormones. The nitrogen gets bound to carbon, and depending on how many carbons are bound to that nitrogen, that'll determine which amino acid is made. And then these become the precursors to protein, and they're stacked together like Legos to make the plant. And every time that you have a bond between carbon and carbon or carbon and nitrogen, that stores energy that originally came in from the sun. So when you start looking at the different products that are around there today, in terms of the plant's world, the plant typically functions and uses as signals or more energetic molecules. You'll see them using what are called L amino acids. And if you look at both of your hands and you hold them up to to your face, you'll see that both of your hands are the same, but they're facing in different directions. So that's how you amino acid, an L or a D amino acid. Now, in terms of nitrogen content, like registered pounds N per product, there really wouldn't be much of a difference. Both of them would cook down to the same amount of nitrogen. But in general, you'll find more of your D amino acids in animal feeds or in directly in fertilizers. And you'll find more of your signaling molecules and more of your highly efficient amino acids in the L form. So plants would respond to L amino acids, and that's one of the things you can look for in a product to make sure you're getting, getting a good quality amino acid package that'll function as more than just NPK.
Okay. So now you talked a little bit about these amino acids. What relationship do they have with crop growth? You said you studied a lot on hormones, plant hormones. That was kind of your big deal. What did you find out with using these amino acids and the growth of that plant and the relationship, the interaction that they would potentially have at different stages of that growth cycle, and what you could do to really enhance specific timings on that?
Well, I'd say that well, in general, they're all extremely efficient ways to deliver nitrogen into a plant. We've seen them over and over again be absorbed into root tips, and some of the smaller ones, if not all of them, have been reported and published to enter directly through stomates and through leaves. So they are going into the plant in single amino acids and small peptides. If you just look at what the specific amino acids are doing, they have some very specific characteristics. Like tryptophan, I said, that's the precursor to auxin. So when the plant's full of tryptophan, that's the amino acid that makes you sleepy after turkey. But that's actually broken down by enzymes in the plant into auxin. Things like arginine. Arginine is directly in the nitrogen metabolism pathway. So it's a storage and transport molecule that allows the plant to move nitrogen to the different growing points. You'll notice the nitrogen deficiencies are always on the older leaves, and nitrogen gets transported to the newer leaves. Well, that'll happen in forms, just in amino acid forms. Some are very well known to act at very low levels and enhance stress tolerance, like glutamic acid, and others would be more partitioned into roots or shoots. Glycine, for instance, is typically more found in the roots, and proline is another really, I guess, a well-known one that's highly connected to drought tolerance. So when the plant's high in proline, you'll find much better drought responses. And then some of the cooler ones, we know sulfur. Sulfur is almost a macronutrient, and it's needed in really high rates. But part of the sulfur metabolism would happen in an amino acid as well, like including the methionine and cysteine. And then some like phenylalanine or isoleucine, typically the aromatics they would be called. These are heavily or involved in the defense and plant immunity responses. So individually, they have really interesting hormone-like activities. Now, the plant itself has main stages of growth, and you can share that chart if you want. But I would say that the three main stages that we could break any crop down would be that early growth, the vegetative growth, and that would start at germination and go right up until the next stage, which would be flowering, when the plant actually starts to mature and its cell division or cell types start to change so that it makes these flowers and fruits. And then finally, a third stage would be this, fruit ripening or grain ripening, and ultimately the maturity of the plant. And as the plant goes through those stages, from seed to seed, it requires different amino acids, different micronutrients, and different hormones. And I think most importantly, it's not necessarily the concentration of any of those ingredients, but it's the ratio of those ingredients to one another. So in the beginning of the stage, you really like to see a cytokinin, auxin, and gibberellin ratio around that 2 to 1 to 1. But then as we transition into more of the flowering and reproductive stages, we really like to see much higher auxins and cytokinins. But gibberellin might be the opposite of that. And then as we get out into that ripening and maturity and ultimately the dry down and death of the plant, that's when you really want to have much higher levels of abscisic acid. And ethylene usually triggers that.
And you're using amino acids to kind of trick these hormone stages, or are you finding some of these hormones within fish products or kelp products per se? What materials are you using to come up with this stuff?
Yeah, I would say both. I don't know. I should probably argue the word trick. But in reality, yes, we are. We're let's say supporting those stages of growth. A good quality amino acid on the market would have it would be digested in a cold temperature process, usually by an enzyme. So it would be predominantly L amino acid forms. So they're very much hormone-like or direct precursors to the hormones. But given that process, a lot of the other things inside the plant are conserved. So yes, you are finding direct hormone concentrations in the amino acid packages that are around, and, well, the good ones anyway. And in those vegetative stages and early stages of growth, in order to get the hormone, nutrient, and amino acid balance that we're looking for, we're typically using an amino acid package that's derived from fish, like a fish hydrolysate. And the aminos in those profiles, or aminograms, is what you'd be asking for. The aminograms there are very supportive of early growth, germination, and that initial vegetative stage of the crop, when that crop's really racing to put on maximum leaves to capture as much carbon as possible. I think where you and I initially crossed paths with your extractors is the oil content of some of the fish hydrolysates that we were working with. So the oil content probably has little effect on the plant itself. Uh, maybe it just makes it a little shinier, but that's what we're finding is really helping promote the soil health. And when you apply your extracts made from the extractors, it's the oil content of these fish hydrolysates. They're protecting the biology that you're applying, and they're actually colonizing a lot better and a lot quicker. And given the oil content of it, if you think of it, there's a diverse, uh, there's a just diverse, lots of diversity of life in the soil, and a lot of your fungi and protists that are helping to cycle the nutrients, they can very easily eat the oil, whereas the bacteria are more living in the water component of the soil, and they're initially repelled. So you do get a selective feeding with some of these fish hydrolysates, particularly the better ones. Now, in terms of then when we start heading into this flowering stage, we prefer to use the amino acid packages that are typically being found in a nice corn hydrolysate. And these corn hydrolysates are also extremely high in cytokinin. Corn is a grass crop, it's full of leaves, and it's actually an artifact in the digestion process of the CSL 896 that we've been working with. And those amino acids are able to promote that cell division that's going on and preparing for the reproductive phase of growth. So let's, for example, like in a corn crop, we'd probably be around V6 to V8 when that's really going on, and that would make a big effect. And then again towards tassel when we're shedding pollen. And we also find that some of the components of those corn hydrolysates are helping to stop direct heat stress in the plant. And that's the time when most crops are very sensitive to it. That would roughly coincide with the beginning of your summers. And then for ripening stages, that's when we'll typically transition into a kelp-based product. And we find the amino acid and hormone profiles of Saccharina japonica hydrolysates are typically very high in abscisic acid that helps to promote uniform ripening and dry down. And really, it helps the plant, it helps stop the plant from cannibalizing its own seed. You've probably worked with some good agronomists out there, and they're always going to be very reluctant to give you a yield estimate based on the number of nodes or number of flowers or whatever it is, what your crop's looking at. And that's because the yield estimate is never right. And usually by the end of the season, you think you had a whole bunch of yield, and your combine tells you a different story. Well, that's because the yield was typically there, but based on the environmental conditions, the plant can suck back those starches or oils that you're growing and reallocate them to new leaves and new roots to stay alive. The plant is ultimately trying to live, and it does that at the expense of its seeds and pods. So, yeah, we like to use a Saccharina japonica. That's what the Kelp 0020 is derived from. and it's full of ABA and even some salicylic acid. And this will help to promote ripening and stop the cannibalization and yield loss that we usually see in September. So if you manage those three stages, you can get the most out of the nutrients and aminos and everything else that you've applied, all your costs. It helps to increase your return on investment in the field.
Back to the fish and the corn. Have you done any research, any work with like soy protein amino acids? They seem to be maybe a cheaper option on the market. I'm just curious your thoughts, versus fish or corn. Some guys may lean towards like a soy protein hydrolysate. What's your thoughts on that? What have you found, if anything, if you've ever worked with those and what you found out with them?
Well, not all, first of all, you have to be careful. Not all amino hydrolysates or liquid hydrolysates are created equal. You really do have to ask yourself some questions. One would be how it's processed. We had that talk with the guys up in Canada, and heat would typically be bad. Heat is going to break down the amino acids into their direct nutrient components. So your carbon will go off as CO2, and your nitrogen will stay in the product as nitrogen. So enzymatic, you're looking for low temperature, you're looking for. You're also looking for L versus D. There's some very economical amino acid mixes on the market that are mostly D. They're more derived from the animal feed business. But in general, the fish hydrolysates are usually your best. They're like the premium of the hydrolysates out there, and you would compare these based on one, their amino acid content. So you can see that it's more than just their nitrogen content, because there's no classification in the United States for amino acids to be registered. We have to register them by their NPK value. So fish also contain the highest concentrations of the largest amino acids. So they have the most carbon to nitrogen bonds or carbon to carbon bonds. So there's a lot more energy conservation that's going on in terms of a fish-based amino acid. And then also with the fish, it's pretty unique to the other hydrolysates, is the fish will range in oil, but most of your fishes are, in general, have a higher oil content than your soybean or corn, and they'll all typically a fish will a pure fish could range in that 80 to 90% L amino acid content, and it would pop back on a feed analysis at probably around 95% crude protein. So fish, I would say, are the premium in the industry, and the fish are still certified, can be certified organic. There's some amino acids out there today that are much easier to mix, but the processing is not organic, so it eliminates that use on some of the more certified acres. Then when we get into I'd say the next range of amino acids, I would put the corn and the soybean in a similar category. The corn will typically have a lower amino acid content. You might find that testing around 50%. But the corn is a lot higher in actual hormones. So the soybean, on the other hand, is mostly glycine. Oh, and that's another thing we should probably discuss is you can rank all these products if you just ask for an aminogram. And a lot of the fish, you'll see some cheaper fish on the market. And if you look at the aminograms, they're really high in glycine. Well, what's the Latin name for soybean? It's glycine max. And we actually named the small amino acid or the soybean plant after the small amino acid because of all of that. So nothing wrong with soybean, but it's about as close to urea as you want to get with a lower nitrogen content. It's
So are you
still
saying
salt free.
the level of glycine in an aminogram would maybe help to determine the origin of the material. Is that what you're saying?
Yeah, we make the PPD 1511. It's a pure fish hydrolysate, and it's literally a technical grade fish hydrolysate that we brought to market. And there's a lot of fish hydrolysates on the market. You'll see them on the internet, or some of them being direct marketed from companies in China. And there's two things that I can always tell if it's pure or not. And one will be the glycine content. And if it gets too high, there's soybean in it.
What
And
would you can just do a range. I mean, what would you consider on the fence or too high
well,
for the glycine level?
I wouldn't say that there's a number that's too high, but I mean, you can smell it. If it doesn't smell terrible, it's probably been blended with something. And the cheaper options to blend it with are going to be soybean, which has the same analysis. So you're not going to reduce the NPK, or the corn, which is a
Just got a
different
much
analysis.
higher glycine percentage.
But yeah, a much higher glycine. In our fish hydrolysate, we rarely glycine is high. It should be the highest of the amino acids in all of your what do you call it? Hydrolysates. But in a fish, you really shouldn't have more than 10%. Whereas in a soybean, you might see in excess of 50% glycine. Yeah, where a corn is usually testing much higher in alanine. So we'll see much higher. That's an indicator I see with a lot of the different corn hydrolysates on the market. Now the corn has more hormones. It has a more balanced NPK. It's the same price as a soybean hydrolysate. So I typically recommend that. And then being a hormone guy, I really do like and I can benefit from all the zeatin that we find in those products. Then
Um,
your kelps are also pretty different. There's a lot of different species of kelps that I've seen. Most of them are brown kelps. Most of them are enzymatically produced, but the different species has shown very different hormone concentrations. We've analyzed countless samples. We've ended up settling on a Saccharina japonica. That's our kelp 0020. Really high in ABA, really high in salicylic acid. Pound for pound, if you were trying to purchase those ingredients, you can purchase them, for pennies on the dollar, as long as you don't mind the all the extra amino acids that come with it in a kelp. But a kelp will typically test in at around 50% amino acid content, and depending on the species, the hormone concentrations can vary dramatically.
Give us a little background on kelps, Rob. Green kelps, brown kelps. You mentioned different species, and they produce, different hormone levels and can do all that. And give us a little bit more information on, obviously the Saccharina japonica species that we represent, but other ones that are out there. And we'll kind of get into maybe times to use those, but just give us some background on kind of some of those kelp materials and what you've seen with them.
Well, they're all generally digested with different enzymes, or their enzymatic digestions, preferably low temperatures, just like the fish, the corn, and the soybean. That'll definitely separate. If you have stuff that's just heat digested or chemical digested, you get mixes of the A and L, or even if they're synthetically produced, you'll get mixtures of A or L and D amino acids. So they're not going to be as active. Most of your kelps are stabilized with a 20% potassium. That's what keeps the bags from eating themselves, because there's enzymes and microorganisms and everything. We settled in on a Saccharina japonica more for the plant's sake than the kelp's sake. We liked it because it was really high in abscisic acid, and it provided a nice tool to stop that late season cannibalization, kind of prevent against yield loss. But I'd say it's rather unique in the industry. I'd say the number one sold kelp that I encounter is an Ascophyllum nodosum. It'll have a similar analysis. It'll have a similar name. They're all going to be called kelp or seaweed or something like that. They're going to be typically, uh, it's a similar amino acid profile if you think about it. Think of where kelps are grown, and even if we're not farming them, but they're usually grown in salt water and they're usually on the top of that salt water. So they're exposed to the sun all the time. They're exposed to that osmotic pressure from the ocean, the salts trying to rip the moisture out of it. And then some of the more low, what do you call it, low depth, or more of the kelps that you would find near beaches, they're exposed to these constant drying and then re-wetting cycles that are usually occurring with the tide. So if you think about it, kelps are like these little chemical factories, and they can't grow deep roots. They can't really do anything to help themselves out. They just have to physiologically make all sorts of amino acids to prevent against stress. So you'll find things like the glutamic acid we were talking about, proline, valine, betaine, and ingredients like that will be pretty common to all the kelps. Now your hormone profiles are starting to vary, with the saccharina being more abscisic and salicylic. Ascophyllum nodosum is typically a very high cytokinin. So it's a different hormone profile. Cytokinin is more like we discussed the effects of the corn steep liquor. So cytokinins, I love them. They're great. They increase cell division. They increase leafiness and tillering. But a cytokinin would likely be one of the last choices if you were looking for ripening. Then there's some minor species on the market. There's an echinchloe. That's similar. It's a very high in cytokinin, similar amino profile. And that's the echinchloe. Then there's a I can always say it wrong, sargassums. And those aren't even considered or classified as kelps. Now you're starting to get into more of the algae markets. So there's actually some companies out of the Southwest that have been growing algae in swimming pools and selling different hydrolysates made from them. And also very high in cytokinin. If you think about what algae is, it's a green cell. So roots, flowers are typically non-existent, and you'll get more kelp dominant or cytokinin dominant species when you think of kelp. But I would look at the species because we're finding that the all kelps are not created equally.
Yeah, that's extremely important. And kind of one of the reasons I gravitated towards everything you were saying was instead of just calling a kelp product and throwing it out there at any point, as a biostimulant per se, if we actually understand what we're doing and what that material is made of and its potential to influence, crop growth at that specific stage, we can do a lot more and be more valuable with that based on the information we have. And that's where I think, yeah, if we can gain this better understanding of the materials we're using, what they're comprised of, and then take that information and figure out the best time to use it, seems like it would be more beneficial to the end user for sure.
Yeah, definitely, definitely. And I like all of these products. Well, maybe with the exception if we're directly targeting soil health. We find not just the timing, like you want to target key timings. So corn's just an easy target because it's so well studied. But in that furrow when the seed's imbibing water and germinating, that is a very key timing. Late herbicide when the kernels are starting to set rows and columns, that is a very key timing. And tassel when the pollen is shedding, the pollen's very sensitive, blanking, the silks. So that's another key timing. So you don't necessarily need high rates of any of these products. I tell people with the fish all the time, if you tried to apply, 200 pounds of nitrogen with a fish hydrolysate like the PPD 1511, I mean, you'd probably kill the plant. It's not to be thought of like a nitrogen, or with the kelp like a potassium. You know, we're typically seeing rates in the quarter pound per acre range if they're going on as a foliar. And maybe it'll get a little higher in a pivot. You might see it up to a pound. But I would seldom use any of these products above a pound to the acre per application. And just try to target very key timings.
Yeah, I was going to say your biggest factor outside of application rate per acre would probably be timing of that cycle and trying to be very specific with which can be tough to do. It's going to rain or maybe it's real dry or things like that are going to happen. Weather patterns are going to happen throughout the season. That's going to kind of dictate what these farmers can do. But at least if we have the understanding at different stages of growth, hitting these crops with amino acids, hormone packages, we can influence that crop growth and do it beneficially.
Yeah, and you just said something too, the environment. You know, all of these hormones that and the amino acids that we're discussing, they're in the plant managing the plant's effect to the or how the plant responds to the environment. So the environment is a key timing. If we go into a drought event, that was a key timing and an application of something like the fish with high proline right during that drought event or right before it could make a dramatic difference in the actual yield from the yield response from those treatments. And then I know we have some farmers laughing in the background because if we could predict things like the weather and heat stress, we probably wouldn't need to farm. And one of the things we like to do, just from a practical standpoint, is we call it low rate multiples. So instead of applying a pound once, I'd much rather apply a quarter pound at 4 different timings. And even with my eyes closed, I'm hedging my bets. And I have a much better chance of hitting the key timing if I don't know it or if I wasn't looking for it. So yeah, I'd say that really applies to the amino acid and hormone products that you'll be working with and using in the field. You know, another key, the baby, a lot of times you really shouldn't, it's hard to chase the response. You know, if you start when the plant is young, you can make meaningful changes as to how it's transcribing its DNA and expressing itself. So if it starts strong, it's going to finish very strong. If you start with these weaker plants and you come in late in the season and you want to do something, you typically have less of a response. So if I had to choose my timings and I had to start cutting things out of my budget, I would front load and I would select the earlier timing almost in almost every case. So like a furrow application is going to give you a much better response than a tassel application if that's the only application that you're using. Just like us, you know, don't skimp on baby food. You'd much rather fast when you're an adult.
Yeah, it's same concepts from the chemistry side of things and focusing on nutrients and hitting things early with specific nutrients, which still is, from my understanding, your thinking is still very important. It's from this biological sense and what that crop is sensing in a hormone level. If we can influence that as well, and still have that chemistry out there and still focus on some of that, we can be twice as productive with twice as little.
Oh, absolutely. And chemistry is another key timing. You know, even in our modern day crops with the genetically modified traits, you know, if you think of a plant, it's billions of cells. And if you look at the way that the plants are developed to be tolerant to different herbicides or even different pesticides, insecticides, fungicides, even though that typically goes on later. But in general, the herbicides, that herbicide, you know, well, without it, you wouldn't get a crop. You need weed control. We know that that's the hardest thing in the world to fight. But the weed control might give you a crop, but it's also stressful on the crop itself. So another really good way to use products like this is tank mixing them directly with herbicides so that you can reduce some of the stress that is farmer imposed on the plant.
Speak more to that. Yeah, that's kind of a newer one for me that came across here a while back was mixing these amino acids with those herbicides. I've heard of, mixing carbon with herbicides, things like that. Maybe touch a little bit about what's actually going on in that whole environment there, whether it's mixing a carbon source or mixing an amino acid with an herbicide. What's the goal with it? What is actually happening out there? I think if we can just have a better understanding of what we're doing when we're trying to implement these methods, I think the better off we'll be in deciding some of
Well,
these different things.
you have to start being pretty careful with some of these applications. Now we've been able to, very successfully reverse, mistaken applications. So some herbicides mode of action, they function by knocking out the synthesis of key amino acids. So the fish, for instance, that's very high in glutamic acid. So we don't like to mix that with those amino acid synthesis inhibitors, or we might be able to reduce weed control. Um, Now, in general, some of them, we like to look for things that are more high in antioxidants when we're actually mixing it with the herbicide. So the general stress response of the plant, well, you have to think about stress. Why does a plant, well, why would this happen? So plant stress is really controlled by a hormone called ethylene. And ethylene is a gas. It's carbon and hydrogen. It's a very small gas, and it'll actually float around the field. So if you're under stress, you'll release ethylene. The ethylene will float over to your neighbor, tell your neighbor that, boy, it's a stressful place. You better start responding to this as well. So it's a contagious hormone. A lot of people are familiar with ethylene. You might have put tomatoes or bananas in a brown bag to ripen, and that's ethylene. That's the ripening hormone. It's generated in a lot of different in a lot of different fruits. Now, for a plant, when a plant grows, its goal is not necessarily to grow into a big tree. Even like the corn plant doesn't want to live forever. What it wants to do is set seeds and then drop those seeds to the ground, and then those seeds will come up next season. So that would be a very successful life cycle for any plant. Just think of the weeds in your field. That's how they grow. That's how the plants have evolved to grow. So one of the best ways to protect yourself is by setting seed and dying. Your seeds will fall into the soil. It'll get worked in there with the rain or whatever it is, and they'll come up, next year, if not many years later. So it's a really good survival strategy. Now, when a plant's under stress, it collects all of its carbon and harvests all of its energy, from the air and the sun. And the enzyme or the actual molecule that does that is called chlorophyll. And chlorophyll is actually a lipid. It's a fat with phosphorus in it. And the fat is going to have a different consistency depending on temperatures, depending on moisture content. Just think of like a stick of butter. If you take it out of the refrigerator, it's going to be very hard to spread on something. And if you put it in a microwave, it's going to be very loose and hard to even pick up with a knife. So that's a dramatic example, but your chlorophyll molecules are exactly like that. So the other cool thing about, or the part of this process that makes it all work is the plant's full of water. I mean, even a drought-stressed plant is going to be close to 90% moisture content. You start getting below 75%. I mean, that's below most people's permanent wilt point where that plant will not recover. So now think of this environment. We have solar energy. We know what that's going to do. It'll give us sunburn, gives us those bad tan lines that you see all the time, and it's relentless. It's coming down all the time on the plant cell, and the way the plant is going to manage all that energy is by capturing it and then taking it and using it to convert CO2 to carbs and CO2 to amino acids and CO2 to fats. So that's how it dissipates its energy. And if the chlorophyll molecule is not the right consistency, a lot of that energy will just bounce off and it'll break off of that energy chain and it'll hit the water around it. And it creates something called hydrogen peroxide inside the cell. And most of us are familiar with hydrogen peroxide. You might have it in the tractor, in your first aid kit. And if you get a cut and you put hydrogen peroxide on your cut, it'll bubble. And that same exact thing is going on in your cell when the plant is under stress. It's not harvesting the energy correctly. It's creating a byproduct, hydrogen peroxide. Hydrogen peroxide is bubbling inside the cell, and that is the signal for the plant to generate stress ethylene. Then the ethylene floats around and it tells all the other plants that it's under stress. So that's basically what's going on. Oh, and then to make matters worse, what is ethylene? It's carbon. That's carbon that could have been starch. It could have been protein. And that carbon is yield loss when it floats away from your field in the form of ethylene. So when you're mixing things like humic or fulvic acid, it's usually fulvic acids they're mixing with it. You'll see like a corn steep product or a kelp-based product when they're mixing those with the herbicides. There's a lot of other ingredients. I was saying they're about 50% amino acid. Well, some of the other ingredients you might find are humic acids, fulvic acids, saponic acid, and all of these will function as antioxidants. So they're actually working to stop the bubbling inside the cell. And that's tricking the plant into thinking everything's okay because with less bubbles, you get less stress ethylene, and then you get less of that, yellow flash, I guess is what we call it in the field after a herbicide application.
Yeah, that visual impact that the farmer can see after they spray that herbicide. They go out there and they see that kind of that yellowing shortly after they apply it, and then it kind of snaps out of it a little bit. And we've visually seen that with using fulvic acid with herbicides and not seeing that so much of that yellowing on the crop after the application.
Yeah, we've been doing that specifically with that CSL 896, very similar. And, right after the application, the plant just kind of sits there for, sometimes up to 4 or 5 days. And the leaves are a little bit limp, a little bit grayer. So the CSL tank mixed with the most herbicides are reducing that completely. And from a more conventional standpoint, we work with another molecule called diformal urea. And this is a reaction product of urea and potassium hydroxide. And it too, it's sucked right into the plant because of that amine nitrogen group. And it's sequestered in those cells right where this process is going on. And when those hydrogen peroxide bubbles hit it, it breaks down into nitrogen and into water, actually. So it helps stop that stress response. But so yeah, you're for something like that, you want to look at something's antioxidant ability. Maybe not its amino acid content. Your other thing with your fulvics and it's never a good idea to cut rates of herbicides, but fulvics do help in leaf absorption, so you might see more active ingredient delivery. That's a good thing. And then now from what we're seeing with these microscopes from a soil health standpoint, one of the biggest enemies to our soil microbiota is really the salt content. I mean, it's a down and dirty test, but the EC rating or the electrical conductivity of the solution that can directly correlate to the amount of diversity that you see in your soil. So a lot of these carbon products, humics, fulvics, even molasses at the right rates, they have a tendency to hide the salt content. Almost like in my world, we make chelates. So we chelate, different nutrients and it protects the charges from the charges in the tank and the charges in the soil and makes it more plant available. Well, your carbon is doing a very similar thing when you apply it. It might not stop the salts. The salts are still there, but they're wrapped. And because of that, they're not directly antagonizing, all those higher orders of life that you might see in the soil.
Yeah, speaking on, the herbicides and things like that, I think the last time we had a talk, oh, there was a question that came up on, I think small grains and desiccation at the end of the growth stage there, and and what you were able to see with using a certain kelp species and how you were able to kind of achieve that, in lieu of using some type of chemical desiccation.
Well, yeah, some guys are using chemical desiccants. It's very common. That was new to me. I came at this I was trained in more horticulture and physiology. So when I first learned that practice, that seemed a little crazy. But it's really common, it depends on your environment. Up where you guys are in Nebraska, getting into the Dakotas, it's probably less needed. But the weather itself, the frost acts as a pretty hardcore desiccant. But when you get down into Texas, like where I am, I mean that's part of your regular practice to desiccate these crops. We first saw it in beans. Soybean is terrible. Well, I guess soybean is great. It depends on how you look at it. But most of today's soybeans are indeterminate, so they'll just keep growing forever until something kills it. And you'll have a hot, dry summer, and the plants are basically shutting down. They're building up high levels of abscisic acid, and it's that abscisic acid. It's protecting it from the drought, and it's causing natural dry down, natural ripening. Then in September, like the third, I think this year it happened in Nebraska, the third week of this of it was the third week of September. The weather kind of cooled off. We got some rain, and all of a sudden, the plant started to shift back to that vegetative hormone profile. Its cytokinin became dominant. It started to make new auxins. We started to see gibberellin in the plant. And the plant made all of these new trifoliates. But that late in the season, it really didn't have roots. It really didn't have nodules. It was just kind of like a dead stick in the now mud. So what the plant has an interesting ability to do is to remobilize the energy from the lower parts of the plant and then turn them back into those components or those Legos that make the new parts of the plant and move them to the tops and create trifoliates. Well, at that time in the season, all of your energy is stored in the actual seeds inside those pods. So a cool trick, get a Sharpie marker. I learned this one in school. And this is what we did in school. We used to draw on plants. But go and write your name on some of those lower pods and then come back. And you'll actually see that the pod that was once full under those conditions can then turn into new leaves and you'll come back to an empty pod. And that's literally yield loss. So a lot of farmers will use just different herbicides to desiccate the crop, and that can get expensive. And then it has variable. There's so many reasons why you might not want to be spraying the herbicides at that time. So what we've been doing is using the Saccharina japonica kelp, the kelp 0020, and we'll go at a high rate. Instead of a quarter pound, we'll go at it at a full pound to the acre. It's very high in abscisic acid just as a byproduct from the extraction of the amino acid process, and that has enough abscisic acid in it to tell that plant to stay ABA dominant. And even though it is starting to make new cytokinins and new gibberellins to become more vegetative and continue growth, yeah, I mentioned earlier, it's the ratio that matters. That external abscisic acid that we're applying through that kelp is enough to keep it ABA dominant, and then it'll continue through the next couple of weeks, ripening and maturing, and then the environment typically changes and it kills off the plant. And now, since we first observed that in the soybeans, yeah, we've done it very successfully now in rye, barley. Corn is super determinant, so it's not always as big of an issue in corn, but we've done it in corn. We've had some success in wheat as well. And then I know it's not the audience for this crowd here, but another one is apples. There's all these new apples on the market that are really cool. They taste better, they look better, but they don't ripen at all. So we've been able to ripen different crops using the same techniques.
Yeah, that is extremely good information, and farmers are all about having tools that they can access and use throughout the growing season. And I think understanding these hormones and the interactions that occur during that growth cycle, and knowing essentially that chart and being able to determine what to use and when to use it and how to use it is an extremely valuable and cheap tool that we can use and be extremely productive with. It's just having that information out there and understanding it and knowing how to obtain some of the materials that may influence some of these hormones.
Well, that chart, I completely agree, and that chart is not theoretical. That chart comes from a patent, and it's an actual model based on growing crops over the course of the season and measuring them at key timings based on growing degree days and just seeing if the
Your
different
screen
parts.
is your screen is shareable. You can pull up one of those that you're referencing.
Oh, nice. I should have been flipping the whole time then.
We don't want to give them too much.
Don't want to. Here's the stress ethylene. This is so where did my bushel go? There's your ethylene flying off, or your bushel flying off. But yeah, these charts are not. Um, this is totally. I mean, this is based on experimental evidence. So as we start from the left to the right, we're typically in a I guess an energy balance where the plant's trying to express its energy and push energy into the environment, and that energy is going to really help to make more leaves, and the leaves are going to help capture carbon and sunlight. And these are the hormones that are typically found in those plants at that time. We're looking for those cytokinins, those auxins, and those gibberellins. Then when we get into this more vegetative, this is the stage that we want cytokinin, auxin, and gib. Then when we get into more of this flowering and reproductive stage, we're really starting to look for those things that are going to help increase the cytokinins of the plant. So this is going to help with your grain and kernel density. We want to bring this up at that time. Then we do want ethylene. You know, no ethylene would mean no AVA and no ripening. You might actually see this if you overuse micronutrients like cobalt in the field. You might see a crop that never ripens. It'll clog your combine. I think it's funny, but farmers don't. But in that stage, like here towards late reproduction, you really want to be careful that you're not generating excess ethylene, because excess ethylene here is going to go off as this molecule, C2H4. This is ethylene, and that is literally at the expense of your grain and your beans and all that other thing. Then when we get up to this final sizing and maturity of the crop, this is when we really want to promote things like abscisic acid and the abscisic acid late in the season. This is going to make sure that we've gotten all of we've sucked in all the energy that we've expressed, and we've filled the grains and the pods to a maximum so that we're harvesting the most of all the input ingredients that we were able to put down in the field that year. And time from seed to harvest, I could put, or we can put a real growing degree calendar on the bottom of this. And then these are
That's what I
real
was
phenotypes.
that's what I
Or
was going to ask you. If you put a growing day calendar at the bottom of this, we have these ebbs and flows of these hormone levels in this plant. And so you can kind of look at this chart and be able to determine when proper timing would be, if we were to put this in a farmer perspective and put growing days underneath this. These ebbs and flows, obviously we have kind of seeding, germination, and then this kind of next fluctuate of gibberellic acid or cytokinin or auxins. What are we looking at? Let's say like a corn crop, pretty common crop. What time of year from a farmer's perspective, so they can kind of understand. Are we kind of seeing this next window of opportunity or ebb and flow in this hormone interaction?
Well, I mean, a farmer can see this. Those farmers are in the field all the time. To some, if you're not in the field, I guess you might miss them, but you can see this in your crop. And if we just go through the crops, so if you're germinating, think of your wheat crop right now. When you start to get to tillering stage of that crop, when you're putting out two, three, four tillers, that is a cytokinin dominant stage of that wheat crop. Then you're going to start to fatten up. You'll actually feel the crop is, I don't know, in grasses, it's pregnant. It would be a pre-boot stage. And if you start to, you know how the grass goes from that soft feeling, you might want to lay down onto it, in a wheat, oat, rye crop or something like that. And then the next day or a couple days later, yeah, it starts to poke your hand when you push down on the top of it. So that would be when it's more auxin dominant. That's actually the heads developing. And then there's that stage in the crop, in these grass crops, including corn, when you can actually see it grow. And that sounds a little stupid, but you can. There's a time period that you were out there yesterday and it might have been knee high. And we go out there, today or tomorrow, and that crop is 6 inches above my knee. The only way a plant can grow that quickly is gibberellin. So your auxins here, these will initiate new cells. So stem cells, the cytokinins will cause all the cell division. So if I had one cell initiated by my auxin, then I would have a cytokinin signal and I would get 2 cells and 4 cells and 8 cells and 16 and 32. And you keep going in this exponential thing, but those cells never fill up with water. They're just dividing and they're very, very dense. And then when the gibberellin signal comes along, it causes all those cells to blow up like little water balloons. And that's when you can actually see the grass grow. And that would be between boot and heading in all of your grass crops. You're probably pushing early July, mid-July in a corn crop. In corn, you're probably at that V6 to V8 stage. And then you'll start to see your first flower form on a crop, on a soybean plant maybe. And that's when this cycle starts again, because the flowers and the developing embryo in that crop, these are all little individuals and they start the cycle again. So you'll get, especially in an indeterminate crop like a soybean, every time you'll get those signals. And then the plant will stop growing. And it's the switch from this exothermic energy release of creating more structure and frame to harvest more sun and more CO2 into this endothermic stage of growth when the plant starts to collect all that CO2 and nitrogen and shoot it down in a source to sink relationship to start bulking my pods or bulking my ears and setting my grain. So that's right here at this stage, right in between vegetative and reproductive growth. And that's when your signals for natural ethylene start to rise on its own. And then that ethylene gets high enough, and that's what induces the abscisic acid to rise. And that'll gradually rise over the course of the rest of the season. It'll actually build up in the seeds themselves. And it's this ABA that keeps those seeds dormant until conditions are right, the ABA breaks down, and the gibberellin starts to express itself again to get them to germinate. So yeah, the farmer can see this. These are cytokinin are very leafy phenotypes. Auxins will have more flowers, more pods. GA will be tall and stretchy. If you plant your seeds too dense, or if you give it too much nitrate nitrogen, you'll see these high GA phenotypes. Ethylene is that angry look that we just talked about after you spray your herbicide and it just kind of sits there mad at you. It's got that dull leaf to it, kind of limp. And ABA is typically a hardened plant, and you might even see a crackly leaf where it might break in the wind if you had too much of the ABA. So, oh yeah, these are real phenotypes. And in season, we'll go around with this chart and even and a microscope. The microscope is new though, and we'll show the farmers where we are on the chart, and then we'll show them the nutrients that they should have in their saps at that time, and then we'll make recommendations based on what we see and try to maximize the balance of this hormone cycle using micronutrients, amino acids, and in some cases, direct hormone applications.
Yeah, speak a little bit about the nutrient interaction in this. I don't want to talk too much on that. Maybe save that for another time. Maybe you got specific nutrients kind of in these different stages here. Talk about the interaction you're seeing with those and what kind of role they can play as well.
Yeah, we like, well, I mean, this was the basis. This is how I started in the industry, was a lot of different micronutrients. And so different micronutrients are needed at different times, and then different nutrients have different characteristics. Some are big, fat, and slow. They could be like calcium, for instance. It can be everywhere in your soil and never in the right place. Um, so calcium typically is it's very supportive of your auxin. It helps to protect auxin and calcium and boron. They're always interacting. So if you don't have one, it could be because of the other one, even if it's there. So calcium and boron are big. In vegetative stages of growth, we like to use a lot of iron, magnesium, manganese, zinc, things like that. That's because, well, magnesium is the direct center of the chlorophyll molecule, so that's going to help with your carbon capture and your efficiency of photosynthesis. And then those other divalent metals like iron, manganese, zinc, and mag, those, if you think of it, they all have 2+ charges, and they're going to work together to help transfer that energy around. So the sun is hitting the plant, it's creating power, so to speak, and then that power can move going through these divalent metals. Then when we get into our more flowering and reproduction, it's really important to make sure you're supporting your auxin. So calcium and boron and zinc are the three most needed nutrients you need to support auxin. So we want to make sure those levels are high. You're typically going to have a lot of zinc because you were following the rules and you were promoting that earlier. But the calcium and boron would be a really good time to hit right at that flowering stage. And then when we get into the actual ripening and sizing and things like that, your boron's very important from that auxin standpoint. When you apply, long story short, but the IAA is a natural plant auxin, indole acetic acid, and boron, it kills or suppresses, let's say it suppresses the enzyme system that regulates the auxin in the plant. If you've ever over applied something like Solubor, you get almost like a 2,4-D response. That's auxin toxicity. And so if the boron is knocking out the enzyme that it's basically eating your active auxins, you can, without any amino acids or any, what do you call it, hormones, you can actually dictate an auxin response just by a boron application. We usually pair boron with molybdenum because molybdenum is the nutrient that supports abscisic acid buildup. So if you can increase your auxins and increase your abscisic acid, you can start to really fatten that plant and drive all the energy from the leaves into the pods or into the ears or into the apples, whatever you might be going at. Copper is a good nutrient for that time too, because copper directly increases the plant's ethylene. So stress ethylene is bad. It's your bushel flying away on you. But, ethylene's not bad. It's the signal to cause ripening as well. So well-timed copper applications can give you a much more uniformity of harvest. And then a lot of crop copper products, they can function directly, I shouldn't say it, but directly as pesticides. And they can help to kill certain diseases, get a leg up on things like tar spot. Uh, phosphorus is phosphorus is another one you don't see me recommending too often. Phosphorus is hot. I mean, it's a very energetic molecule. So a lot of phosphorus has a tendency to increase ABA. So I use it when I need ABA, but I don't use it indiscriminately. And you'll notice on my chart here, I have no nitrate. Nitrate, I'm always recommending urea. Urea would be my favorite nitrogen source, amine-based nitrogen. Hence me stumbling into the amino acid world. Those are just amines that are connected to carbon. So nitrate, I shouldn't pick on it. Nitrate is a highly soluble, highly bioactive nitrogen. The plants take it right up and they use it. But my problem with nitrate is it increases your gibberellic acid. So in most of the systems that I work with, the gibberellic acid will increase the height of the plant. It'll make your plants tall and skinny.
Makes it grow
And
really fast and doesn't really do much of anything else. So
yeah, yeah. So even in your lawn. I mean, if you want to do a quick experiment while you're home, put urea on the left half and a nitrate source on your right, and you'll be out there cursing me because you're going to have to mow the nitrate a lot more. But yeah, we typically want our crop plants to be short and fat, and they're going to that'll promote a lot more fruiting, a lot more of it's just a more productive plant. So we kind of use all these things in combination. And now you probably heard enough you start to think, oh boy, why have I been putting zinc in my furrow mix? Or, well, this is why. So you really are a lot of what we discussed on this call today, you're already doing. And you just probably didn't know the mechanisms behind it.
yeah, like I said, that's I think the most important thing is just knowing what we're doing and why we're doing it. And I think that's what you can kind of help connect the dots to is, both on the chemistry side and this, biological concept of it is why are we doing this and what is it going to do to this crop when we need it to do it? And then that's a lot of the answers that we kind of look to help growers to find and not only help them, do some of this, but provide them the information so that they can somewhat know what they're gonna do out there is gonna perform for them.
Yeah, and you want to be working with the crop, especially now when efficiency is more and more important now than it ever was in history. The prices of all the inputs are up, and now too, a lot of the environments and the states that we're working around in, the regulations are also up. I know in some of my territories, we have to write nutrient management plans to justify why you're applying these things. So, as you go, you might be able to look at your own programs and see that you're using some things that you need at those timings, other things you might be using at the wrong timing, and maybe you want to apply that earlier or later than you have in the past. And some things you might find that you'd want to eliminate it all. We in a hot, dry year, you might not need to promote things like ripening. You might want to do the opposite, and promote things that increase your vegetative growth.
Yeah, no, that's good. As we look to wrap this up, Rob, in hopes to provide some more information and maybe go into some more in-depth analysis on specific things. And I think maybe the next topic of discussion that I would maybe like to like to talk about and maybe have some growers listen in on is how can we take this information and make it useful, but how is this going to increase production or save us on other inputs that we may not need? Kind of that interaction there, talking about NP and Ks and then how we can maybe look at alternatives that are going to increase crop growth, increase crop production, and do that more efficiently. And then yeah, kind of hone in more on your charts there relating to those different stages and then what we want to focus on in that specific stage. And obviously with planting coming up for most of the US here in 2 months, probably starting, give or take, 2 or 3 months. Um, Planting is going to be an important time. So talking about um, what guys should be doing at that time, what are important factors to look at, and kind of your intel on some of that. Those are kind of some topics that I would like to touch on, maybe going forward into another video here in the future, and maybe you can just touch on some of that. But I think we've got a really good start here.
Yeah, definitely. And I mean, I think that's great. If you look at the actual stages of the crop going left to right, we have our germination, planting time, vegetative stage, then we'll jump into a flowering stage, and then the ripening stage, and we can time that with the calendar so it's appropriate and on everybody's mind. Planting time is really important. There's a
Stressful
lot of
time.
great
A
seed
stressful
out there,
time.
and there's a lot of great seed. And I don't want to sound cliche, but there's the old question that I ask the guys in the field, but when is the highest your yield will ever be? And that's in the bag. When that seed is in the bin or the bag, when you buy it, that's the highest yield potential you will ever have. So it's not really about increasing your yield as the season goes along. It's more about taking advantage of some of these theories and practice and preserving the yield that you paid for in that bag of seed.
Yeah.
So if we start in the furrow, there's quite a few ways that we can right off the bat make a really big difference. And seed treatments, that's probably the cheapest way for a farmer to get uniform coverage of every single plant in the field. In furrow applications, if you support the little baby, think about that. I have a few hobbies, right? But I like bonsai trees, so I'll make these little bonsai trees when I have time. And if I tried to take an oak tree and shape it to my will, I would need a chainsaw. I would need a tremendous amount of energy and money and time. But if I start with the acorn, it's very easy for me to grow that plant into whatever I want it to be. So your corn, bean, wheat crops are no different. So I think that'd be a good one. We could talk about in-furrow applications and side dresses, the micronutrients that you want to put in those, the amino acid profile you want to put in those, ways to increase that biological diversity right in that furrow. And we can get our audience up to a really good start. See if we can finish the race strong.
Yeah. Yeah. And I think to tie into that too, as guys probably already have most of their plan put together, but want to, kind of finalize some of that plan is, um, some of the, these are going to be different, products per se, programs, practices that these guys, just a different understanding that these guys would potentially look at and understanding some of that, but understanding maybe the excess that they're putting out there from a year to year and where you can really hone in on what's going to be useful and what's not going to be useful. And like you ran a TAPS program here recently and
Oh
had some really good
yeah,
results
taps.
and
We
you
were
were
too
able
unconventional
to
for
grow
the
that corn
award.
crop for, a small amount of outside nitrogen and be able to hone in on this chart and use this information productively and not lose a lot of yield, but have a really good ROI. And so talking about things like that is really I think what guys are going to want to hear as well. I think a lot of guys will take this information in, but having a call to action, having a plan in place, and how they can implement some of this stuff is hugely important to just the information as well. How can they take the information and use it on their farm?
Yeah, absolutely. And if you have an abundance of nitrogen and you just want to put that on the ground, the reason you need so much is because it is so inefficient. If you start to target these things to specific timings when the plant needed and when it's taken up, you can literally use a fraction. It's probably a bad agronomic recommendation to tell guys to quit using NPK, but it's not uncommon to reduce those rates by 75% if you're doing it, at the exact right timings. And you
Mhm.
don't even have to be that crazy. You can be off a little bit, just reduce it by 50%. But you'd be amazed how little it takes to grow a crop. I mean, we're growing commercially comparable crops at a half the nitrogen just by applying the nitrogen at the right time, in the right place, using supportive micronutrients, using the amino acids to stimulate growth, and partnering that with the fertilizer so that you signal the plant to pop roots. Those roots then suck up the fertilizer you put down. It's all about increasing the efficiency of farming. It's not about increasing yield. Plants grow yield on their own. We have to be more efficient with our practices. When inputs were, cheap, it didn't matter as much. You just over applied, and then the plant would free choice feed, just like the cows. But now that, these ROIs on these fields are, I mean, in some case, break even. You have to be much more careful what you apply, and you have to make stricter decisions.
Yeah, efficiency. We're all about that, and having that the right information is very key to knowing what's going to be most efficient for you. I think that's a great stopping point for this video as we look forward to another one and being able to get some of this information out there. And yeah, I think this was a good stopping point, Ron.
Yeah, definitely. Well,
So,
this was, I think, a really good place to start, and I look forward to a whole line of these. We'll do a bunch of different episodes, and we can focus on key timings. And I could always be found at Agmerica, and I do make field visits, so I look forward to working with everybody out there.
yep, that sounds good. Thanks, Rob.